All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So, once again, thanks a lot to Paul Borg for talking about the stats. This was our first session on that. We will keep improving on it. What I wanted to do, even when this is a chit chat, I wanted to do a very important discussion. And this discussion is going to go off of a Twitter thread. But I think once you see that thread, you would feel that this is a credible Twitter thread and can be used reliably, or at least more reliably than some other Twitter messages. The message in this one is very interesting. They have done a research, and I'll show it to you, and I'll read it with you, and I'll draw it with you as well. But the basic summary of this is the following. This is beautiful. They are saying that they are seeing the spike protein of the Omicron to be not as efficient as Delta in causing severe disease dynamics. So when Delta becomes or causes severity, it has a bunch of things that it does. For example, it does syncytia formation. Syncytia formation is, imagine these are the two cells, and these respiratory system cells have little hair on them, so or little uh, appendages on them, very tiny things on them, and the SARS-CoV-2 Delta was able to fuse them, so it would cause them to become united and become units, and that is what syncytia is. Syncytia means a unit, so it would fuse the cells, and that is how the severity, that is one parameter of the severity. And this researchers found that Omicron cannot do that, which is very interesting, which is lucky for us. It cannot do that. Similarly, they saw that Delta was able to more efficiently enter into the cells by getting better primed. Remember TMPRSS2 that primes the spike protein? There is a little protease that is on our surfaces. And when the spike protein arrives, the protease kind of cleaves it. I'll draw it for you. Omicron doesn't use that phenomena very efficiently. So then the question would still remain, if Omicron spike is not efficient enough to allow it to become primed fast, then how the heck is it entering the cells faster and causing more infection? And maybe that may be wrong. So it may be that it is not entering the cells faster. So we'll see. Let's look at this beautiful set of study excerpts that this research lab has put forward. So ready to join brains and look at this together. I really loved it. And as much as there are so many folks who are actually not comfortable at this time when the information is evolving and they say, hey, stop, only talk once you have better information or more reliable information or studies or RCTs. I love this evolution in information. So let's see, this is drbeen.com. Then here is the tag, and thank you to the cool bean who tagged it. This is Gupta Lab, and Gupta Lab is Ravi Gupta is a professor of clinical microbiology, Department of Medicine, University of Cambridge. He was named in the Time 100 most influential globally in 2020. This is the Cambridge Institute of Therapeutic Immunology and Infectious Disease, Cambridge. And we'll talk about this as well, split GFP, in a second. I want to go over this. So get ready. I'm going to draw a little and discuss it. So here is what they're saying. They're saying, and look, December 17, it is hot off the press, and 12:33 p.m., sharing some potentially significant findings related to Omicron given the current situation. First of all, huge thanks to the team working flat out, Bo Meng and our collaborators, both in G2P, J2P, along with systems virology. Findings are follows. So check out the findings. Omicron spike protein mediates deficient cell entry. That's very interesting. So we have to continue to start thinking as well that we were thinking that this virus enters the cell faster. Here we are looking at Omicron spike protein mediates deficient cell entry. And again, this is an in vitro study. and is inefficiently cleaved compared to delta spike. So let me just draw this part of it. What they're saying is that, and my apologies, I don't mean that you, if you read it, you cannot interpret it. Not at all. I like drawing. So I would think about everything in terms of drawing. So um, apologies freehand. There is no assumption that you need this illustration. So here, let's say this is a spike virus, and here is a spike protein. What they're saying is that, remember, we have TMPRSS2. That is a protein. This is TMPRSS2. This protein has a little scissor. It's a protease. That means it cuts the proteins, and it would prime S1 and S2. Remember, it kind of cleaves them away. It separates them. That causes the S1 portion to get stuck here on the ACE2, while the S2 portion, once it has become separated from the S1, then like a arrow, it it bolts and fuses with the cell membrane of our cell. Once it fuses, then it pulls the remaining virus and fuses the membrane of the virus with the membrane of our cell. That causes the RNA in the virus to go into the cell, and then the rest is history. It goes in, replicates, creates more viruses, then they come out, and then they do the same to the next cells. What the researchers are saying is, and of course you can understand that here, this priming of the S1 protein or the spike protein is important for the SARS-CoV-2 viruses to enter our cells. They're saying that Omicron's priming is difficult, and I would assume because of mutations. So because the priming is difficult, it is not able to enter the cells with that much of the ease as Delta. So Omicron spike protein mediates deficient cell entry and is inefficiently cleaved compared to Delta spike. We tested viral entry mediated by wild type, that is a Wuhan, Delta and Omicron spikes using a pseudotype virus system infecting primary 3D lung alveolar organoids. So the whole thing is uh, artificial, it is in vitro. So what they've done is they have a virus. Let's say you take a human coronavirus and you stick the SARS-CoV-2 spike on it, or you take another hollowed out virus and you stick a spike protein on it. So this becomes a fake SARS-CoV-2 because now we can check its binding capability. Then you take a tissue cell that you have built in the lab, which has lung cells in it. So it kind of looks like lung tissue. So that is what they're saying, 3D organoid, ORG ANOIDS. So I was correct with the A. I thought it was O. So organoid means something that looks like an organ. So this structure looks like a lung from a tissue or the cell composition. So then they put these viruses onto this structure. And they put Delta there, and they put Omicron there, and they put Wuhan type or wild type. And then they see how are these getting cleaved, how are they entering the cell, and what is their behavior. So what have they seen? So now imagine these little fake particles that have these spike proteins attached on them. This one here, the black one, is the wild type. So this is a particle. Imagine a small microscopic, electron microscopic ball, tennis ball, tiny ball, on which you have attached the little needles, which are spike proteins. So Wuhan type spike proteins, this is here infectivity. So let's say 10 raised power 4. Then Delta type spike protein, 10 raised power 5. And then Omicron type spike protein, 10 raised power 2. Less infectivity. No spike, just that little tennis ball. Why did they have a tennis ball which didn't have any spike? They wanted to make sure that there is a control available as well, and it's not just those things are just entering the cells regardless of the spike, and then we are just thinking it is spike. So that is why the control, you can call this no spike as a control. So what they saw here is the spike of the Omicron is not the most friendly spike for it. And please realize that at the end of the day, virus, as it is evolving, it is going to try to become more human or host friendly. So we are saying it is not friendly to it, but really it is friendly for us, that it would do less damage to us. Anyways, this is one. Now, this stays in contrast to, let's say, the study yesterday from Hong Kong University, where we said, or the researcher said, that they saw the virus entering the cells with 70 times more avidity. So if it cannot bind to ACE2 correctly, then compared to delta and wild type, then how is it entering the cells 70 times faster? Both are correct. Both researchers, it's not that one or the other is lying. We have to figure out what are those mechanisms. So that is the part of evolution of the knowledge, that you're seeing one study here that says, hey, it infects less. The other study says it infects 70 times more. Now the question is, what is happening? And then the researchers are going to go and find that as well. I really love researchers who do these things. Okay, continuing. Next point. Omicron spike protein induces relatively poor cell-to-cell fusion compared to wild type and delta. We expressed spike in cells stably expressing split GFP so that green signal could be measured over time upon cell-to-cell fusion and syncytia formation. The difference is significant. So let me explain this part as well. Again, no disrespect meant here. It is just that I would love to draw this part. So here is what happens. What they're saying is, let's say this is lung epithelial cell. Let me make that in blue. This is a lung surface cell, epithelial cell. Epithelial cells in the lung, those who are in the healthcare, they know that these cells are sitting on a basement membrane. They have these little spikes on top of them, not spikes. They have projections. And then their nuclei are near the bottom. They look like pseudo-stratified epithelium, correct? So some are small, some are big, and so some have nuclei down here, some have nuclei higher. So now this is becoming a histology lecture. So they said in the older variants, what is observed, and this was a common research, it's not something that was hidden, that is that when the virus particles, they attach to these projections, what they do is they cause the cells, these projections, to fuse with each other. And when they will fuse, the cell would start fusing, and we will end up with a syncytium or units, meaning there are multiple cells that just got fused as a unit. So then the cells react to this kind of, this is like if we take a bunch of people and just tie them together. So they would react and they'll say, what the heck? And so then the immune response would occur and the cytokine storms would start occurring and the immune system would just start going mad for this behavior. What they're saying is that with Omicron spike, when they infected these cells with Omicron, these projections, these cells did not fuse with each other with as good of efficiency as wild type and delta. And this, they're saying, is an important marker or indicator of less severe disease. Because part of more severe disease is the lower lung tissue damage. And if this virus cannot fuse them, it cannot weld them together, it cannot bind them together, then it would not cause those extra immune system responses which are occurring because the syncytia formation is going on, because the cell destruction is happening, because the cells are becoming fused. When that happens, so healthcare workers here or students, when that happens in macrophages, we call them mega karyocytes. Sorry, we call them, what is that? Those cells called? Uh, Big macrophages with multiple nuclei, multinuclear giant cells. We call them giant cells, right? So giant cells are where macrophages have fused together. Here we're talking about epithelial cells fusing together. So for us, generally, the response here is when a virus comes in and fuses our cells, our system goes mad, causes cytokine storm in some people, and then even kills them. Omicron doesn't do it, or doesn't do it as avidly. That means lower respiratory tract infections are not as severe, which is a good news. This observation was there yesterday from the Hong Kong University's lab as well, where they said that lower respiratory or the lower lung tissue, not lower, the lung tissue infection was 10 times lesser than Delta and the wild type. And they said that may be an important reason for less severe disease. And here we have Gupta labs from Cambridge also showing us why this may be. So they are showing that the virus itself is not able to cause fusions and cause damage. Very, very interesting. So this is the second point. So if you see here, the blue, the black, and the green. The green one is Omicron. And if you see blue and black, that is the Delta and the wild type, they are making syncytias. They are fusing cells together. They're welding them together. They're tying them together. They're generating clusters to which immune system would say, what the heck have you just done? And start attacking that area. And Omicron is standing there not doing much. The SARS-CoV-2 parents said to the SARS-CoV-2 children, go and tie the cells together and their bodies would kill them. And this Omicron is not tying them together a lot. So syncytia formation is less. So that is the second point, very interesting point. Third, what does this all mean? Efficient infection of lung cells could correlate with severity of lung disease. So they're saying that it is possible. And this is how researchers speak. They would not say this is how it is and that's it, this is the fact. They would always say this could be a possibility. So they're saying the virus's efficiency in causing lung infection is or could be an important indicator of the severity. Syncytia or fused cells are often seen in respiratory tissues taken following severe disease. So they're saying those folks who have had severe disease, if you take their tissue, you would see that a lot of cells of their tissues have these syncytia formations. So there is multiple cells that have become fused together, and they are in this syncytium, in this unit. And this is usually present in the severe cases. Now, is it because it causes severity, or is it an outcome of the severity? We don't know, but it is seen in them. And it turns out in vitro, Delta and wild type is able to do it, but Omicron is not with as much efficiency. See here. So then they say Delta was very good at both infection and syncytia formation. Both things, Delta was great, good. In contrast to Omicron, further work is needed. So Omicron is actually losing out to Delta in terms of causing the damage. It is winning out to Delta in terms of transmissibility. The question is, how do we now balance out the message yesterday that we have a greater load, 70 times more load in the bronchial airways versus here? Now, it is possible that the cells over here may be behaving differently compared to the bronchial tissue. Again, that was also in vitro. They call it ex vivo. That is, you take cells from the living humans and then do test on them. And here they had organoid cells. These are specialized cells that are there for testing. It's interesting, very interesting. Then they say, we also tested how well antibodies from vaccinated individuals neutralized Omicron versus Delta. So now what they did was they took people who had antibodies and they took their serum. And of course, the serum had the antibodies in them. Then they put Delta in those serum and they put Omicron in those serum to see how well those antibodies attacked these viruses. So again, I'll just draw it because I think I should I should be in fourth grade. So here, so let's say this is serum from patient A, some patient. And this serum has antibodies against Delta because this person was vaccinated against Delta or against SARS-CoV-2. And now when you put Delta in here, these antibodies are going to try to attach with the Delta spike protein and try to neutralize it. So they are saying that here we took these serum from various people and we put those serums that had antibodies and we put these viruses in them. So I'm going to make Omicron in green. So they said we put those viruses and then we wanted to see how well these antibodies performed, these antibodies. So let's see, what did they see? We found that Omicron was poorly neutralized after 2 doses of messenger RNA. So if you see this here, this is Pfizer vaccine, and look at Omicron on this right side. 1 month post second dose. So this is a person who had his second dose and then had 1 month after, and they requested the serum, or the serum was present somewhere, and they added Omicron to that serum, and you can see this is the outcome. Here, 6 months post second dose. This is the outcome. Again, I think you know my position that 6 months after the second dose is usually antibodies have waned. If you look at Pfizer's CEO says our antibodies wane anyways. So if there are no active cells here, just the serum, then adding the virus will not activate the memory cells which would start producing the antibodies. So this is just whatever antibodies are there or not there is what you get in the serum. So 6 months later, so somebody who had second dose and then 6 months after, they took that serum and put Omicron in it. And this is the result. Then they asked another person who, let's say a month ago, had their booster. And they said, can we please have your serum? Or they had the serum somewhere. And then they put Omicron to it. And if you see, there was a better result for neutralization. Of course, I'll keep saying this. The boosters would ramp up the immune system. The sleeping cells are going to become active and they're going to start making antibodies. So the person would have more antibodies. So anyways, good observation. And it is just in line with all the immunological mechanisms. Here is the Delta. And what we saw was, this is the interesting thing. 1 month after, if you see, Delta was better neutralized by whatever quantity of antibodies were there. 6 months after, post second dose, a similar thing where Omicron was less neutralized, Delta was still better neutralized. Again, whatever quantity of antibodies were there, tiny bit or not. And 1 month post the booster for Delta, almost very similar to the Delta, the Omicron. So they said, we also tested how well antibodies from vaccinated individuals neutralized Omicron versus Delta. Now, if we take this as a fact, let's say we agree with the researchers and we say, yes, this is what is happening. You give a booster, or somebody who got 2 doses and very recently, let's say instead of 6 months after the second dose, 1 month after the second dose. If we say that the antibodies are able to neutralize Omicron, then this concept that somehow Omicron has escaped the immunity is not a correct concept. We would probably say that efficacy has reduced, but we can't say it has escaped. And why do I make this statement? Remember the South Africa study of 12 samples only, that made the whole world go in a spin, which said the vaccine efficacy is gone or the immune evasion has occurred? Or remember Oxford study a couple of days ago that we saw that had 43 samples? I mentioned these 43 sample, 22 on one vaccine and 21 on the other, because when these are 12 or 43 samples, if these are studies for some other drug, then those studies will be thrown out and people talking about them will be shamed and and made fun of to talk about those small studies. But this second study as well caused a very large reaction from the mainstream media, from the healthcare authorities to say, look where we are at. However, I want to put this with that to say, if booster causes the antibodies, which are the antibodies to wild type, and these start equally neutralizing Omicron like Delta, then we cannot claim that Delta Omicron has escaped. And making this statement that only T cells would help but the antibodies will not help, here in this serum, there is no memory cell or T cell helping. It is just the antibodies. So either we agree with this or we say, you know what, for my purposes, I want to agree or I want to feel that the virus has escaped. And for that purpose, I want to dismiss this study and say the others are correct. And then some folks would say, I would rather dismiss the other ones to say they have smaller data and go with this one. Again, this study has its own, or this Twitter thread has its own limits. This is an in vitro study. This is done on pseudoparticles. This is done with organoids instead of the actual tissue and so on. So this has its own issues. These have their own. The right way to look at it at this time is to say there is data coming from many directions. Data are not sufficiently large to be able to make conclusions, but it is interesting that we are getting these kind of data. So if you ask me that, hey, do you think now from South Africa and Oxford study that Omicron has escaped? No. As you can see here, that Omicron, and as you can continue to see people saying that the severe cases are not happening, it is so funny for me. Just let me make one more point. When UK, and I really love UK's stats and data and dynamics of the data. I love it. I think Israel and UK have done the whole world a favor with the data they have provided. But the commentary and the, just like I am making a statement, statements I don't really very much, I'm not a big fan of those statements. So one of the statements that has been out very commonly yesterday, day before yesterday, that, well, the South Africa is 90% protected. That is why they have less severe cases. Good. Let's take that as well as a fact. If we take that as a fact, South Africa's vaccine is only 25 to 30%. Where did the remaining 50 to 60% come from? They are, of course, then infected and recovered, which South Africa says, no, that's not true. But let's say that is true. Then aren't we in this one swoop, at one fell swoop, aren't we agreeing that natural immunity has such huge benefit against Omicron that the cases are mild? And aren't we then agreeing that these antibodies against Delta are able to work against Omicron as well? So anyways, meaning there is a lot of varying information coming up and varying statements coming up as well. We have to keep ourselves sane during this process. So here is a good graph to look at. So we also tested how well antibodies from vaccinated individuals neutralized Omicron versus Delta. We found that Omicron was poorly neutralized after 2 doses of mRNA or adenovirus vectored vaccine compared to Delta, but that the third dose mRNA vaccine rescued this at an early time point. So if you see here, this is the third dose or the booster, and that almost brought the neutralization of Omicron versus Delta almost to the same level. So would you, after seeing this, let's say for the time being, we agree that this is a correct study and it is reproducible, it is reliable. Looking at this study, would you say that Omicron has escaped? Okay, I want to continue. Then they say, in summary, let me back up. Efficacy may have reduced, but the antibodies that booster produced are not going to be some other antibodies. They are similar antibodies against the wild type. In summary, this work suggests that Omicron does appear to have become more immune evasive. That is based on these two first dose, second dose results, I think. But that properties associated with disease progression may be attenuated to some extent. So that property of having a priming capability, getting primed by the TMPRSS2 or creating syncytia, these are attenuated. The significant growth of Omicron nevertheless represent a major public health challenge. Now, for me, the question left is the following. I understand syncytia formation. That is fine. So it doesn't cause much severity. It is becoming more and more upper respiratory tract infection. Upper respiratory tract type infection type, and it is sparing the lower lung tissues better. So that all is fine. But if it cannot infect as well, then we have a problem. So my request to Gupta Lab will be one additional test, and that would be the cells from the upper respiratory tracts, or specifically from bronchi, to be put separately and seen what kind of infectivity is there. Maybe, just like children, I said this yesterday as well, maybe just like children, this change has brought Omicron to a point that it can work very well with the upper respiratory tract system cells and less well with the lungs. And that would be a good news.